Hello students, today we are going to continue with biology chapter number 11, enhancement of food production. Okay, this is the second lecture of this chapter, second part of this chapter. Okay, so in our last lecture we are studying the chapter enhancement of food production. Okay, so today is the second lecture. In our last lecture, we have started this chapter. We have covered the introduction part of the chapter. Then we studied about the plant breeding definitions. Then we have studied the objectives of plant breeding. Why we are doing the process of plant breeding. Okay. Today, we are going to study the topic of hybridization in crop plants and its technique, how hybridization is done. Okay, why we want to do the hybridization. Okay, so and how we are doing the hybridization. Okay, so is the food production per annum produced per annum in our country sufficient to meet the ever increasing population? So the answer from you, I expect, is a big no. Okay, so I expect that you all will answer a big no, that food production per annum in our country is not sufficient to feed the ever increasing population. So what can be done? Thus, there is a need to enhance our agriculture efficiency. Okay. So there is a need to enhance our agricultural efficiency. We need to increase our food production. Okay. So how can we increase our food production? The most important technique that we can use to increase the food production is hybridization. Let us define the process of hybridization. It is the process of developing plants and animals having some desired qualities like a high yield, disease resistance, tolerance to climate, etc. So when I say hybridization, the definition is as follows. It is the process of developing plants and animals. We can do the development of plants as well as animals. And in those plants and animals, we can get the desired qualities like high yield, disease resistance and tolerance to climate. This process is called as hybridization. Okay. Next is types of hybridization. There are two types of hybridization. First one is intervarietal hybridization and second one is distant hybridization. Okay, first one is intervarietal hybridization and second one is distant hybridization. So we are going to study one by one. Okay, there is no need to study the types in detail, but just the definitions we have to study. Okay, we have to just study the definitions of all this. So intervarietal is again divided into two, that is intraspecific and interspecific, and distant hybridization is divided into two, that is intrageneric and intergeneric. Okay, so starting with the definition, first one, intervarietal hybridization. What do you mean by intervarietal? Varietal means variations, varieties. Okay, intervarieties. Okay, now intervarietal means between two varieties of same species. The hybridization will be involved between two varieties that belongs to the same species. The parents involved in this hybridization belong to same species but are having two different varieties. Okay, there are two different varieties of same species. Example is given here, crossing of two varieties of wheat or other crops. Okay, next is intravarietal. Okay, intravarietal, intervarietal. Intravarietal means the crosses are made between the plants of same variety. Okay, crosses are made between the plants of same variety. Okay, intervarietal means we have seen that the crosses are made between the plants belonging to two different varieties. So intravarietal, intervarietal. Is it clear? Intra means of same variety and inter means of two different varieties. Crosses will be made between two different varieties. Next is interspecific. Interspecific. Specific here refers to species. Okay. So interspecific means the crosses are made between two different species of the same genus. Okay. Interspecific means the crosses are made between two different species of the same genus. There are two species but their genus is same. That crossing is known as interspecific. Okay. Next one is intergeneric. Okay, intergeneric hybridization. This means that the crosses are made between two individuals of different genera belonging to the same family. Okay, it means the crosses made between two individuals and those two individuals are of different genera, means they are having different genus, but they both belong to the same family. Okay. So what is it going to happen? It produces the resistant varieties as well as a desirable combination of all characters. Okay. Example is brassica. When crossed with Raphanus, it will give us Raphanus brassica, Raphanobrassica. Next is distant hybridization. When I say distant hybridization, it means the parents involved belongs to different species of the same genus or of different genera. I can simply say that as parental plants are distantly related, parents are distantly related, such crosses are also called as wide or distant crosses. Okay, in this parents are distantly related to each other. Okay. It means they belong to different species of the same genus or of different genus. Then, next is interspecific and intergeneric hybrids are seldom to occur in the nature. Means the most occurring crossing are interspecific as well as intergeneric hybrid. Okay, they occur too seldom in the nature. Okay, now moving on to the important part of the topic, that is steps in plant breeding, steps in hybridization. The question can be main important steps in plant breeding program or main steps in hybridization. Techniques of hybridization I've told already. Okay, so how the hybridization technique is done? How is plant breeding technique done? So one by one, there are 5 steps that we are going to study in detail. First step is collection of variability. Okay, first step is collection of variability. Second step is evaluation and selection of parents. Third step is crossbreeding of selected parents. Fourth step is selection and testing of superior recombinant. And the fifth step is testing, release and commercialization of new cultivars. Each step we are going to study in detail one by one. Okay. First step, collection of variability. It is very simple. Collection of variability. Variability means varieties of species and collection means we are going to collect the varieties of species. Okay, variations are the differences found among the offsprings with regard to their parents of the same species. It means different varieties of plants we are going to collect. Okay, such genetic variations are heritable and are useful in their selection. Variations are very useful in the selection. Okay, so first step, what we are going to do? We are going to collect the different varieties of species. Okay. And the entire collection having all the diverse alleles for all the genes in a given crop is called as germplasm. When we are going to collect the different wild varieties, species and their close relatives, all this collection of diverse alleles of all genes in a particular crop is called as germplasm. Okay, germplasm conservation can be done in two ways. Okay, germplasm, first step is clear. What we are going to do? We are going to collect all the different varieties of species, and this collection of different varieties of species is called as germplasm. This germplasm conservation can be done in two ways, that is in situ conservation and ex situ conservation. In situ conservation means it can be done with the help of forest and natural reserves, and ex situ conservation means it can be done with the help of botanical gardens and seed banks. Okay, the germplasm that we are collecting, we have to conserve it. So for the conservation, there are two ways, in situ and ex situ. In situ means with the help of forest and natural reserves, we can store the seeds. Okay, and ex situ means with the help of botanical gardens and seed banks, we are going to store the species. That means we are going to conserve the germplasm. The varieties of species that we have collected, we will store that. Okay. Second step is evaluation and selection of parents. When I say evaluation, it means checking. Okay. Now we have collected all the varieties. Okay. Second step, what we are going to do? Evaluation and selection of parents. In that collection of germplasm, from that germplasm collection, we are going to select the parents. This is the very important step and essential step. The collected germplasm is evaluated. We can say we will do the screening of the germplasm, and then after screening, after testing, we will identify the plants with desirable characters. Okay, we will identify the plants with desirable characters, and then this selected parents are means they are self-pollinated for 3 to 4 generations to make them pure or homozygous. We are going to selfing. Okay, we are going to do the selfing of them. Okay, then why we are going to do selfing? So that to make sure that only pure lines are selected, multiplied and used in the further process of hybridization. Okay. Now after this, the third step is cross hybridization among the selected parents. The variety showing maximum desirable features. Now in this hybridization, the selected parents, we have selected the parents. In those selected parents, we are going to do the crossing. Okay. After selfing, what we are going to do? The crossing. Now crossing will be between two parents. So the variety showing maximum desirable features is selected as a female parent, and the other one as a male parent which lacks good characters, but those good characters are found in the recurrent parent, that is female parent. So one parent is having all the desirable characters and the other parent is lacking some of the good characters. So what we are going to do? We are going to cross them. Hybridization is the crossing of two selected parents differing in one or more characters. As I have told you, that female parent will be having all the good characters and the male parent will be lacking some of the characters. Now the hybridization is going to make the combination of useful characters and possible, which are usually scattered in the different varieties. Okay, it is also useful in exploitation and utilization of hybrid vigor. How are we going to do the process of hybridization? That we will study now. Okay, definition is hybridization is crossing of two selective parents differing in one or more characters from each other. Now there are some steps of hybridization that we are going to study in detail. Okay, in your textbook it is given in very short, but we have to study this in detail. It is very much necessary to study this first, then only you will understand that one line that is written in textbook. Okay, Hybridization, steps of hybridization is emasculation, bagging and tagging, dusting of desired pollen grain, rebagging of female flower. Okay, emasculation means what? The process of emasculation means the removal of anther from the bisexual flower of selected mother plant. Okay, emasculation means removal of anther, that means removal of male reproductive part from the selected mother plant. Okay, in the early condition, bud condition, before the maturation, that is called as emasculation. Emasculation is done with the help of forceps or hot water. Okay, emasculation is not required for selected mother plant with unisexual flower, but if the flower is bisexual, we need to do the process of emasculation. Okay, means simply you have to remember that emasculation means removal of anthers, removal of male reproductive part from the selected female parent. Okay, then bagging and tagging. Bagging and tagging means what? The emasculated flower is covered with the help of bags made by butter paper to avoid the interference of other pollen grains. That is called as bagging. That means we will cover that flower so that no other pollen grains will come and pollinate this flower. That is called as bagging. And tagging means just we will add a label on that flower so that we will remember the name of the plant and when that process of emasculation was done. Okay, that is called as tagging. Next is dusting of desired pollen grain. Now once after the emasculation is done, we have only female flower, female plant. So on that female plant, what we are going to do? We are going to dust the pollen grains of the male flower that we want. Okay, the matured pollen grain from the selected male plant are dusted on the stigma of the flower of the selected female plant. And then the rebagging of female flower, that means the female flower is rebagged and allowed to develop into fruit. Okay, after the rebagging, the plant pollination is followed by this fruit formation and seed formation. The seeds thus obtained represents the hybrid generation. Okay, the seeds that obtained are going to represent the hybrid generation. The hybrid F1 progeny is selected and evaluated for the desired combinations of characters. The progeny that we are going to get after this step are then going to be evaluated for the desired combinations of characters. Okay. Next is the fourth step, that is selection and testing of superior recombinant. The F1 hybrid that we have got, now from the third step, we are going to use that here. For plants showing superiority over both the parents and having high hybrid vigor are selected. Okay, the progenies obtained after hybridization are observed for desirable combination of characters and these progenies are then used. Such hybrids are then selfed for few generations to make them homozygous for said desirable characters till there is a state of uniformity, so that the characters will not segregate further. Okay, now these plants that we are going to get are going to be again selfed. After hybridization, we are going to, the recombinant superior variety that we have got, we are going to self-pollinate that again, so that homozygosity will be maintained. Okay. Fifth step is testing, release and commercialization of new cultivars. The newly selected lines are evaluated for their productivity and other features like disease resistant, crop resistant, quantity of the crops, etc. Okay. Then the evaluation is done by growing these plants under the controlled condition. First, they are grown in the research field under the controlled conditions such as fertilizer application, irrigation, crop management, etc. After that, they are grown in the different agroclimatic zones, agricultural zones in natural conditions. And then finally the variety is released as a new variety for the use of farmers. Okay, and that variety is again tested by IARI, that is Indian Agricultural Research Institute, and then they are sent into the market with a new name, new variety, and with a new name with the permission of government. Okay, this has led to the Green Revolution. Development of high yield varieties of wheat and rice in 1960s by plant breeding technique has increased the food production, and that is known as Green Revolution. Okay, now we will study about some of the Indian hybrid crops in detail. Some of the crops which have developed because of hybridization, which has shown tremendous increase in its production because of hybridization. First is wheat and rice. Increase in wheat and rice production was done by introducing semi-dwarf varieties, and there was a tremendous increase in both wheat and rice production. The scientist named as Norman E. Borlaug developed the semi-dwarf varieties of wheat. This scientist Norman E. Borlaug is also known as father of Green Revolution, means he has done too much work in this field, so he was declared as the father of Green Revolution. Okay, some of the varieties we are going to study. The most important here, you have to remember the names of the semi-dwarf varieties. Semi-dwarf varieties of wheat are Sonalika and Kalyan Sona. Okay, Sonalika and Kalyan Sona. And the semi-dwarf varieties of rice are Jaya and Ratna, and even Padma is there. Okay, Jaya, Padma and Ratna. Now, these semi-dwarf varieties of rice were taken from IR8. Okay, IR8 means International Rice Research Institute. See here it is written IR8 here. Okay, so it is International Rice Research Institute and Taichung Native are these two places from where the rice for semi-dwarf varieties were taken and then they were introduced in India. Okay. Next crop is sugarcane. There are two types of sugarcane. First one is Saccharum barberi, that is grown in North India and had poor sugar content and yield. Saccharum barberi species are having very poor sugar content and yield in it. And Saccharum officinarum is the second one, and it is the native of South India. It is grown in South India and it has thicker stem, high sugar content, but the problem is that they don't grow in North India. If we take them to the North India, they are not going to grow in that place. So what can be done with the help of hybridization? These two varieties were crossed with each other to get the desirable qualities of both. Okay, high sugar content and thick stem and ability to grow in North India. So after the crossing, the sugar species obtained after crosses showed high sugar content, thick stem, and they have shown the ability to grow in North India also. Okay, now there are some hybrid varieties of sugarcane which you need to remember is CO421. Since this sugar hybrid varieties were developed in Coimbatore, okay, it was developed in Coimbatore in Tamil Nadu, okay, so it is given the name as CO421. CO represents Coimbatore, 421 is the name of the species. So CO419, CO4205, CO453, CO740. These are some of the hybrid varieties of sugarcane, okay, that were developed in Coimbatore. Next crop is millets. Okay, millets means what? Millet is generally the term for grasses yielding small seeded edible grain. They are small seeded edible grains. Several varieties of jowar, maize, bajra have been developed. The varieties of maize are Ganga 3, jowar are CO12 and bajra is Nifed that have been successfully developed in India. These varieties are high yielding and resistance to water stress. So these all are the crops that have developed because of the technique of hybridization. Okay, so again we will repeat once the steps of hybridization. First step is collection of variability from the germplasm bank, then evaluation of selection of parents, selection of parents from with different qualities. Selected parents selfed 3 to 4 generations to make them homozygous or true breeding. Then identification of parents as a male parent donor and female parent recurrent. Collection of pollen grains from the flower of the male parent. Removal of stamen from the flower of the female parent. And then artificial cross-pollination by using pollen grains collected from the male parent. After that, bagging, tagging of the emasculated flowers of the female parent. Then the development of fruit and seed representing F1 hybrid generation. Then selection and testing of F1 hybrid for the combination of desirable characters. And then field trial for yield, that is productivity. Then we are going to do the testing and the release of new variety with the new name. So this all are the steps of hybridization which are very important, and you need to remember this. Okay, I hope you all have understood this topic. Okay, so thank you. We will continue this chapter in the next lecture again. Take care.